I see you like just share this and then just. Is that easier? Cool. Not many students. Anytime after 9.
Hello. Hello.
Yeah, right. Oh, I could. Oh, right. I can do it. Okay. I mean, if while you're sharing, if people. Yeah, that's true. Hey, you're the first person here. Yeah, you made it. Yeah, it is. Thank you. That's not mine. That's Heather. No. That's what I had some of. I had some of my lunch already. Yeah, it should be like lunch something. There's so much of big food. Yeah. Yeah. And that big. Oh, cool. Lunch. Hi, Don. I see that you're there. Can you hear this? I don't know if you can. Oh, here we go. Oh. Great. Thanks, Tom. Yeah, that'd be a good idea.
Like they're living abroad. So how long is your presentation? You know, more or less?
It's like 37, 40 minutes.
Okay, that's
I
good.
mean, I'll be timing myself enough to try to get close to.
Do you want me to check the clock or something?
Sure, maybe once the quarter to one.
Okay. 12:45.
Maybe I'll grab it till one. The next class. Some classes.
Yeah, it's different. So I should tell you what time do you want me to do? Like 12:50.
I'll give you the
10 minutes. Okay. Okay. Hey, so you want to use this, right?
Yeah, I can use.
Okay. I'll tell you. How do we know? Okay, so you use like that maybe?
Yeah, I can say.
Okay. So hi everyone, I think we get started. It's okay. It's yes, let's get started. Okay, there is no camera, they cannot see us, right? Okay. Okay, and so hi everyone. It's my pleasure to introduce Drew Harner. Drew is a PhD candidate in the horticulture program, probably one of the last few students in the horticulture program. So Drew and Meredith, yes. So Drew joined Penn State in 2016 after graduating with a bachelor in plant science, viticulture and enology, and a minor in English at Cornell University. So he joined our program as a master student, and then after completing his master, he decided to stay for a PhD, and that was 2019, right? Still working in our group in viticulture. So Drew will graduate this summer, and this is not his exit seminar. It's just, you know, an update on his progress or some of the research that he has been doing on grapevine lanternfly interaction. And I'm glad, you know, Drew is working on this topic because it's a challenge and it's a timely issue for our grape growers. So with this, I'll let you start. So we're not going to be late. Okay. Thank you.
So if anyone can't hear me at any point, please just throw something in the chat and we can see. But so thanks, Michaela, for the introduction, and thanks to everyone who is here. I'm excited to be presenting some results from a couple of my dissertation chapters looking at grapevine responses to phloem feeding by the spotted lanternfly. So the spotted lanternfly, or SLF as we typically abbreviate it, is a phloem-feeding plant hopper, originally native to East Asia, so like China and Vietnam. And I'm sure many of you have heard it by now, but maybe you've not seen it before. And these are what adult spotted lanternflies look like in a vineyard setting. And especially on the right side with some of those, with a lot of the insects on the trunks, that's typically reflective of the type of infestations that we've been seeing here in Pennsylvania. So I say that it's a phloem feeding insect. So most, insects like caterpillars and things like that directly eat the leaves as when they feed on plants. But in terms of spotted lanternflies and other sap feeding insects, they have this little stylet that you see on the left side of the screen that they essentially use to tap into the phloem or where the sap is in the plants, kind of like a mosquito. And that's how they get their nutrients and resources from the plants themselves. And these insects typically feed on the shoots and the trunks of grapevines. So the spotted lanternfly is not native to North America, but it was first discovered in North America in Southeast Pennsylvania in 2014. And since then, its population range has expanded pretty rapidly throughout the eastern US. And this map is from, on the right side, is from March 28th. And you can see all the blue counties are where there's currently documented populations of spotted lanternflies. And the red outlines are quarantine or state level, I'm sorry, county or state level quarantines. So as you can see, it's pretty much present in almost all of New Jersey at this point and more than half of Pennsylvania, including Center County here at Penn State. So we'll probably be seeing them this season. So worldwide, it's a generalist insect, meaning that it can feed on 103 different plant species. But here in North America, only 56 are actually documented food sources, with the most important food hosts being tree of heaven, Ailanthus altissima, and unfortunately grapevines. They really seem to like grapevines. And as a result of that, Pennsylvania wine grape growers have reported reduced yield, fruit quality, and vine health following multiple years of successive heavy infestation events. And when we think about spotted lanternflies within the vineyard, they can spend their entire life cycle in the vineyard. They typically emerge from their eggs early in the season, so on the left side of the graph or left side of the slide. And they have 4 juvenile instar phases before they reach their adult life stage at late in the season. But typically in the vineyard at this time of year, they're actually really easy for growers to manage because they're less mobile than when they're adults. And additionally, growers can simply manage their populations using various types of insecticides, either contact insecticides that will just kill the lanternflies outright, or systemic insecticides that the plant takes up. And then when the lanternflies feed on the plants, they then die. So as a result, growers can manage them a little bit easier in the early part of the season compared to the end of the season, in which there's some more complications. First, adult lanternflies can jump and glide and fly. They develop much increased mobility compared to the juvenile stages. They typically move around the environment more in groups, so the chance of an infestation coming into a vineyard is simply higher. And in addition to that, they're feeding on the vines during an extremely important time for both the growers and the grapevines. And that's because the adult stages emerge during when the fruit are ripening, and will then feed on the vine while the fruit is trying to ripen, and we want quality fruit as wine grape growers. But additionally, because of the close proximity to harvest, growers often can't apply the same insecticides that they can use earlier in the season. So they can't use those systemic insecticides because then we potentially get insecticides in the fruit, and that's not good. So instead, they're relying on contact insecticides, which can kill and knock back populations within the vineyard. But typically what happens, within a couple days up to a week, more adult lanternflies will move back into the vineyard. So there's no guarantee of preventing future incursions. And what happens is that growers get this kind of weekly cyclical reinfestation event of vineyards. So it's really difficult for growers to manage at this time. And it's important to note too that there's very, very little to almost no literature on spotted lanternfly management. We're not operating with really any guidelines here. So growers typically, when they see a lanternfly, they spray. So a lot of growers are spraying much more than they have in the past, which is not good economically, and also has led to kind of explosions of secondary insect pests following these insecticide applications. So it's really difficult at this time of the year to manage them. And then when you think about what's happening in the grapevine at this point in the season, as I mentioned, fruit are ripening. It's very important, but also the vines are allocating resources to various perennial tissues. So when I say perennial tissues, I mean really anything that's woody. So like the trunks, the woody shoots, as well as the roots. And the vines are actively allocating any carbon that they absorb through photosynthesis to the fruit to ripen the fruit. We want ripe fruit that gives us good wine. And then also they're allocating carbon to these perennial tissues for both metabolic processes during this time of the season, but also for long-term growth. Because grapevines are perennial plants. We don't harvest the whole plant at the end of the season. We just harvest the fruit, and then we manage the plant to live, multiple years. But in order to do that, we need to manage these concentrations of resources in the perennial tissues. So in addition to carbon, they also do that with nitrogen. Nitrogen is absorbed through the roots and then allocated throughout the canopy in the plant. And then the grapevines are allocating that to the fruit, which is important. Nitrogen in the fruit is important for fermentation dynamics and yeast. Yeast need nitrogen, so it's also very important for wine quality. And then grapevines are allocating nitrogen to these perennial tissues again for long-term storage. So the problem then becomes, in terms of the spotted lanternfly, when you add an additional stress onto these vines, particularly an insect that can ingest large amounts of sap that is carbon and nitrogen rich, what is really happening to the resource allocation throughout the vines? Are these resources not actually going to the vines or to the fruit, but actually going to the lanternflies instead? And especially, and as I mentioned before, there's very little research on spotted lanternflies. And so we're kind of operating in the dark in some aspects, because we don't really know what exactly they can do to the vines, but we can infer various potential effects from other sap-feeding insects. There's a ton of sap-feeding insects in the world. Many are very common, like leafhoppers, aphids, mealybugs, some of which I show on the left. And they're also well studied in agricultural and perennial plant systems. And some general themes that emerged that maybe we can use to try to hypothesize or propose what spotted lanternflies may be doing to the plants are that these insects tend to reduce carbon assimilation. So the amount of carbon actually being, brought into the plant. They also have been shown to alter plant resource allocation. So when I say that, when these aphids or other insects feed on the plant, various studies have shown that they can shift how the plant is moving resources around, either to benefit the insect, meaning that the plant is allocating resources towards the insect, or moving resources away from the insect to prevent resource consumption by the pest. And additionally, a lot of these insects are known for transmitting plant pathogens. This is a big threat in terms of grapevines, because there's a variety of plant pathogens that aren't present in the northern US that are present elsewhere, and we're worried about the spotted lanternfly being able to transmit these. So there's various things that we can read the literature and think about when thinking about the lanternfly. And especially this is important because, as I mentioned, there's such little literature out there that we don't really know what the mechanisms are that are leading to what growers are reporting, this reduced yield, this reduced plant viability. As you can see on the left and the right, these growers are having some pretty substantial infestations with a lot of lanternflies, and they don't really necessarily know how to manage them. At what level of population size should they start managing them? And this is something that's fairly common through insect pest management and perennial crop systems, through like integrative pest management and basically identifying specific levels or population sizes at which growers should start managing them to reduce negative effects on their crop. So this kind of outlines the framework for what my research that I'll be presenting today is about. And really, to start thinking about the main objectives that we targeted, we really had the question at the basic level: do spotted lanternflies affect resource allocation? When you put spotted lanternflies on the vines, are they affecting the movement and the allocation of resources to both above ground tissues, so like the fruit, especially, as well as above ground perennial tissues in the trunks and shoots, but also what about the roots? The roots are where the grapevines allocate a lot of their winter dormancy reserves, and these reserves are especially important for next year's growth. And then what about when you have a ton of lanternflies? You know, not only do they affect resource allocation, but what happens when you really just have infestation population densities that are, you know, really extreme? What happens then? Can the grapevines withstand it? And then a second objective, as I mentioned before, many sap-feeding insects can affect carbon assimilation in plants. And this is important to understand whether this happens in grapevines, because if so, then this is essentially impacting the amount of carbon that the plant is able to absorb, which then could have downstream effects on plant health, as well as fruit ripening. And also because carbon assimilation and leaf gas exchange is so responsive to sap-feeding insects, we really thought that maybe this would give us a clue as to what's happening in the vine physiologically in response to this feeding. And then, as I mentioned, you know, grapevines are perennial plants. We need them. We'd really rather not be replanting every single year. So we need to make sure, or we need to better understand, how does this intensive feeding in a single season affect the growth or the health of the vine in the year after? Because again, growers have been reporting reduced plant viability and plant mortality in some cases. So we want to understand what may be driving that. So these three objectives really kind of formed the main part of one of the chapters of my dissertation. So to investigate these objectives, we set up a study in 2019 and 2020. The first two different experiments. The first experiment was a field experiment in which we caged 16 Riesling vines in both years. And you can see photos of the cages on the left side of the screen. And we exposed these vines to a variety of different population densities of spotted lanternflies, from 0 to 15 lanternflies per shoot, or essentially 0 to 200 per vine. And we basically would just go into the environment, collect a certain number, introduce them to the cages, and then see, you know, do various measurements there. And it's important to note that to try to replicate this repeated cyclical kind of infestation cycles that growers are seeing, we did that here. And so we would go out and collect the insects, introduce them to the cages after 4 or 5 days, remove them from the cages, give the vines maybe, you know, 3 or 4 days to have no infestation, and then reintroduce lanternflies. And again, we did this to kind of mimic the insecticide driven reductions in population, but then reinfestation that growers are seeing. Then in 2020, we did a potted vine experiment using a different grapevine variety, actually with totally different species, Marquette vines. We had 12 of them. And for this, we just exposed half of the vines to 80 lanternflies per vine, which was a very high amount, and the other half to 0, so a control. And we did this in order to validate the results we saw in the field experiment, as well as do some additional measurements that I'm not going to be reporting on here. So to measure these, you know, the resource allocation component of the study, we looked specifically at the end of season starch and soluble sugar concentrations in perennial tissues. So again, those woody stems and those woody roots. And by looking at the end of season concentrations, this would give us a good idea of what the cumulative effects of spotted lanternfly feeding would have on these concentrations, as well as these, you know, carbon reserves that the grapevines had going into the winter, into the following year. And then we also looked at the fruit. In this, we measured total soluble solids, which is a typical just measurement and proxy we use for ripeness. And again, the fruit is ultimately what grape growers are concerned about and what winemakers want to know about. So we wanted to see what was happening to the carbon allocation there. Then in terms of the nitrogen, again, in the same woody tissues, the stems and the roots, we measured end of season total nitrogen. And then earlier in the season, around harvest, we looked at the total nitrogen of the leaves before leaf senescence, just to see if any nitrogen depletion was happening in the canopy. And then again, we looked at nitrogen in the fruit. And we measured this in fruit in terms of yeast assimilable nitrogen. And this is simply the pool of nitrogen that yeast can use to ferment grape juice into wine. It's very important for winemakers. Then in terms of carbon assimilation measurements, we have a little portable gas exchange analyzer that we use in the field. There's a photo of me using it on the bottom right. And essentially, this is a single point in time measurement. So it can only do one leaf at a time. And we measured 3 leaves per vine for all 16 vines across both years at a series of different time points, usually at the end of the infestation cycles, to see how spider lanternflies were affecting the carbon assimilation. And as much as we would like to do more, we would have liked to do more measurements. This is a fairly time-intensive process, so you can really only do so many leaves in a given day. So we were kind of limited here to these sequential point-in-time measurements, but still, they gave us an indication of what the spider lanternflies were doing to the carbon assimilation rates of the upper canopy leaves. So then to transition to results, I'm just going to kind of show a snapshot of our main results that really are kind of the emerging theme of what these couple of years of research are showing. And the first is that in terms of that resource allocation, we see a very, very strong end of season reduction of starch in the roots. So in this in this graph, on the y-axis is starch in milligrams of starch per gram of tissue. And on the x-axis is just the spotted lanternfly density, starting from 0 or no lanternflies, all the way up to 15. And each of these dots indicates a measurement from an individual vine. And really what we see is that even though the spotted lanternflies feed on the canopy above ground, they never touch the roots. They're having very strong effects on the roots and really reducing the amount of starch that the grapevines have in the root systems going into dormancy. And coupled with that, we see a slight increase in soluble sugars. So whereas starch is more of like a reserve type, storage type of carbon, the soluble sugars is a bit more labile and are a carbon form that can be moved around the vine easier. And interestingly, we see an increase here. And this is not what we had expected to see, but grapevines tend to be pretty plastic and able to adapt to various stresses. And in previous research, some researchers have shown that in situations of carbon limitation or carbon stress, grapevines will actually reallocate the carbon in their roots to above ground sinks. And we think potentially that's what's happening here, that not only are the spotted lanternflies potentially reducing the total amount of carbon that's going to the roots, mostly because they're potentially just absorbing it all themselves because they're in the canopy, or potentially there's just a reduced amount of carbon available to the grapevines, which I'll talk about in a minute. It looks like potentially that the roots are reallocating some of that carbon above ground. And interestingly, we see this in the second experiment in the potted vines too. So this is results from the Marquette vines, again, an entirely different grapevine species. And what we're seeing here is that regardless of the cultivar or the variety, we're seeing consistent effects on below ground starch and soluble sugars. So this is, again, just a graph showing starch between the control vines and then the vines exposed to spotted lanternflies. And this is about a 70% reduction in starch. And then we look at soluble sugars. Again, you see this slight increase in soluble sugars, suggesting that potentially there's a remobilization or retranslocation of carbon from the roots back up to the canopy. And we have a variety of hypotheses that may try to explain what's happening here. One is that, again, there may just be a reduced pool of carbon such that there's not enough carbon really even going down to the roots by the grapes. The grapes aren't translocating that carbon down to the roots. It's possible that the grapevines are mounting some type of defense response, and that's really metabolically intensive. And it's likely that the grapevines are reallocating that carbon, those sugars, to then use for defense responses above ground. And the interesting part about that is that when you look at the above ground tissues, so as I mentioned, we also looked at carbon allocation above ground, and in these stems, or in the stem tissues, and on the left is the stem phloem or bark. So we essentially just removed the phloem and the bark because lanternflies are phloem feeding. and tried to see if we saw effects on that, and then also effects on the interior of the stem, the xylem and the wood. And in both of those, you see an increase in starch. And what this is suggesting is that, again, coupled with this reduction of carbon allocation to the roots, there's an increase in the stems. And this is a very common stress response in other perennial plants when they're exposed to phloem-based stress. And it looks like it's happening here. And that for whatever reason, the grapevines are just really accumulating starch in the stems in response to increasing lanternfly infestation. So then when you think about rather the allocation, but the amount of carbon actually entering the system, so that carbon assimilation I was talking about, we also see a decrease in the amount of carbon being assimilated by the leaves. And these graphs I'll show you is just carbon assimilation on the y-axis, so the amount of actually carbon being photosynthesized, and then again, spotted lanternfly density on the x-axis. And after, and again, this is data from 2020. And what we saw in 2019 is the same as this. So after the first cycle of feeding, 6 days of feeding, we saw a linear decrease in carbon assimilation that was there, but not that strong. But then after an additional 14 days of feeding, after cycle 2, we saw an even stronger reduction in carbon assimilation in the vines that were exposed to allotted spotted lanternflies. And then by the time we got to the end of the experiment, or an additional 8 days of feeding for a total of 28 days of feeding, it appears that as these vines were exposed to increasing densities of spotted lanternflies, the carbon assimilation rates were just going down very fast and pretty much staying at a level after these 28 days of feeding. And not only did we see this, but we also saw reductions in foliar transpiration and stomatal conductance, meaning that with more feeding, and especially in cases of more exposure to higher population densities, the leaves were just shutting down and not photosynthesizing, which is not good because then that means whatever carbon is being removed by the spotted lanternflies is most likely not being replaced by further assimilation. And then to speak quickly about nitrogen, we did see some reductions and effects in terms of the total nitrogen at the end of the season, but these were really proportionally less compared to the carbon resources, and there were really limited effects on above ground tissues. But it's important to note that we just looked at total nitrogen and no other aspects of nitrogen metabolism. So further research would really like to kind of target what's happening in terms of nitrogen dynamics in the vine. But it looks like within a given season, it's really a story of carbon limitation. So then, as I mentioned before, being grapevines or perennial plants, what about next year, kind of year after effects in the growing season immediately following infestation? And what we saw, our results from the field experiment were rather inconclusive due to various environmental problems that popped up, mainly spring frost, that just really made it difficult to measure any effects. But in the potted vines, we had 2 vines that had complete bud mortality, meaning that they didn't grow at all the following year. And in those vines, they had root starch concentrations below 1%. So it looks like in these vines exposed to high spotted lanternflies with their limited root systems and real limited soil volumes, they just really depleted, the spotted lanternflies depleted almost all their carbon, resulting potentially in this mortality. But we didn't see any lanternfly-induced mortality in our field study with the Riesling. So even though we saw this in the potted vines, the vines in the vineyard really seemed fine. So just may have been like a potted vine effect, but still worth noting. So the main conclusions from this section of my study was that heavy spotted lanternfly phloem feeding progressively reduced carbon assimilation, so the amount of carbon going into the vine, and then altered that resource allocation in terms of carbon and nitrogen throughout the vine. And then in terms of the largest effects, it was really the roots that bore the brunt of this altered resource allocation. And the depletion of root carbon that we saw is most likely involved in bud mortality. And then important to note, as I mentioned, we were trying to identify at what population density we really saw substantial effects to then relate to growers. And it really seemed like the strongest effects were in vines exposed to populations higher than 100 lanternfly per vine. And also we exposed these vines for like 28 days continuously to spotted lanternflies. And most growers would not do that. They would spray after a day or so. So we really tried to test the limits of what these grapevines could withstand. So while this study gave us some good foundational insights, we had a lot of kind of remaining questions, and limitations of the study. So what happens in the grapevines when lanternfly are no longer feeding? As I mentioned, we removed lanternflies for a couple of days before introducing them. We don't know per se what's happening in this in between time, and if grapevines can respond differently or try to replace what's lost to the lanternfly, or what's happening. Then also, do the whole vine responses match what's happening in the leaves? In the terms of the carbon assimilation data, we only measured what was happening in single leaves and not the entire canopy, but the whole canopy's response may be different relative to the leaves. So it's important to understand that as well. And then also, does the spotted lanternfly damage the phloem? When you have 200 lanternflies on a vine for 200 days, feeding on the same tissues, it's most likely that they're incurring substantial damage to this tissue. And that's something that we wanted to explore too. So the second chapter of this study was really looking and exploring at the xylem and phloem dynamics of these grapevines that we infested with spotted lanternflies, specifically in regards to what's happening in the xylem. So the amount of water being moved in the xylem transpiration, as well as what's happening in the phloem where the lanternflies are feeding. Both not only, when lanternflies are on the vine, but then what happens when they're removed? Do we see an increase in physiological activity to compensate? Do we not? Is it permanently inhibited? These are the questions we were asking. And then what about repeated feeding? After 6 infestation cycles, were the vines even functional? Just basic questions about the vine physiology in terms of the xylem and the phloem. And to investigate this, we installed a variety of continuous measurement tools. So before the experiment, we installed sap flow sensors that we just put right on the trunk. You can see what the sensor looks like at the top of the screen. And then on the right is a photo from the installation, as well as here. And these sensors really measure any movement of sap in the stem. That's largely going to be transpiration or the water moving up through the stem, but they also can pick up to some degree what fluid is moving in between tissues as well. So this gives us a continuous whole vine kind of integrated measurement of what's happening physiologically. But then to look at more tissue-based responses like the xylem and the phloem, we installed dendrometers. And an example of the dendrometer is in the photo in the middle. And then the sensors you can kind of see in the photos on the right. They're very hard to photograph, but essentially they just measured the displacement of not the displacement, rather the contraction and expansion of the xylem and phloem tissues in a given day. And so as the grapevines are transpiring, some tissues will swell, some will shrink. And by measuring these with these dendrometers, we can really get like real time, down to the second insights into what's happening when the vines are stressed. So to jump right into it, I'll first show you my sap flow data. So this is data from 2019, and this graph on the right is showing average sap flow rates through the experiment. So on the y-axis is sap flow and the milliliters of sap per meter squared leaf area per hour. So hourly rates. And on the the x-axis is the dates of our experiment. So every date where there's a dotted line, that is when we had an infestation cycle and lanternflies were on the vine. And then the means, or the little box and whiskers that are not on dotted lines, are those in between those cycles. And I simply just average multiple days of data to get those points. And what we see first is that sap flow, even in vines that aren't infested by lanternflies, decrease as the season goes on. This is late in the season. As you can see on the dates, it's August, September, October. Pretty much by the end of the experiment, the vines were beginning to senesce. So sap flow rates and vine transpiration is just going down naturally. But when you look at the vines that are exposed to spotted lanternflies, the low treatments are about 40 lanternflies per vine. Didn't really show much of a difference until the very end. But again, at this point, this is early to mid-October. So we're not really necessarily sure there's a real biological implication of this difference. But as you begin to look at the other treatments, in the medium vines, so about 100 lanternflies per vine, we started to see more, beginning to have reductions in sap flow across the whole experiment. And again, we did see significant differences in some of the off cycles, which we don't really know how to explain. But just again, the overall trend, meaning that sap flow is beginning to decrease with more lanternflies. And then you really see it in the high lanternfly treatments. So 200 lanternflies on a given vine, and you really see a strong reduction in this green line beginning around cycle 2. So about 9 days of feeding, sap flow is starting to decrease, and it stays decreased throughout the rest of the experiment. So not only, were we seeing these reductions in carbon assimilation in the first half of the study, but we're also seeing these sort of substantial and persistent decreases and reductions in sap flow throughout the experiment. And again, this only gives us, a whole vine idea of what's happening. So, when we're thinking about in terms of the tissue, oh, I'm sorry, and I should mention before I get to that point, again, significant reduction starting in cycle 2 through the end. To look at those specific tissue effects again, we looked at the dendrometer data, and on the right axis or the right side of the screen, you can just see a little diagram showing what the sensors look like when they're on the plant. And we essentially just carved out the phloem and the bark and put the sensor right on the xylem. So we're measuring the exact xylem traits. And the graph on the left just shows it's a box plot of what we call the amplitude. So the amplitude is simply calculated by the max, kind of radius of the stem minus the minimum. And this gives us essentially an index for the total activity. Like xylem activity for the day is how we interpret it. This data set's really kind of difficult to work with because it's a data type that is new to us. So we're trying various ways to kind of summarize the data, and using the daily amplitude is one of them. And what you see is that there's a range of amplitude values for the control, but then when you look at the lanternflies or add more lanternflies to the vine, this range of amplitude values begins to shrink. And the way to interpret this is that as you have more lanternflies on the vine, it looks like the xylem activity is reduced or slowing compared to vines with less lanternflies or no lanternflies. And this is something that we need to explore further, but preliminarily it does look like xylem flow is reduced, which again matches that sap flow data. And then in terms of the phloem, again, the lanternflies are phloem feeding. We want to know what's happening to the actual phloem tissues themselves. And we got this phloem data by the other dendrometer that's positioned on the bark. You just simply subtract those measurements from the xylem dendrometer, you get kind of the phloem and non-xylem tissues movements. And when you look at the amplitude values here, what you see is an increase. So there's an increase in daily amplitude with increasing lanternflies. And this is more difficult to interpret because, as I mentioned before at the xylem, as you get a reduction in amplitude, you get a reduction in activity, and we would have expected a potentially reduced activity in the phloem because these sensors are at the base of the trunk and the lanternflies are feeding on the top of the vine. And we're seeing this increase instead. So, we have a lot of questions about this. Does this mean that the lanternflies are actually increasing the phloem activity of sap up to, of sugars and sap reallocating from the roots back up to the canopy, or is something else happening? And this is something we're working actively with one of our collaborators. It's a very difficult data set, but we're excited about where it may lead us. So then to kind of summarize that part, the early evidence of what we see from this continuous kind of measurement data set is that we can say for sure that spotted lanternflies are reducing xylem flow. In terms of the effects on the phloem flow, that's still something we're investigating. And again, it looks like these reductions that we see in the xylem and with sap flow are persisting following repeated infestations, and even in this, in between no infestation phase. And as I mentioned, we're concerned about whether potentially spotted lanternflies are damaging the phloem. And related to this is a question of, well, just what degree are these vascular effects that we're seeing, the xylem and phloem effects? Are they a function of tissue damage? And someone tipped us off a couple of years ago saying that they noticed in some maple trees that were infested with lanternflies that there were these marks beneath the bark. So we decided to look at our grape stems. So these are images of grape stems with the bark removed. And what you see are these little lesions or striations. We have like 8 different names for them. We don't really know what to call them. But we suppose that these were potentially damaged to the phloem by the lanternfly, because when the lanternflies are exploring the vine and trying to feed, they'll often probe the vine and taste the phloem sap. And then once they found a spot where they like, they'll feed. But in doing so, they likely damage the phloem repeatedly. And that's what we think we're seeing here. So to further visualize this, we submitted some stems to Ben Hall, who works for Lasers for Innovative Solutions, to kind of visualize these stem tissues via laser ablation. So this is just simply a cross-section of a 1-year-old grape stem. You can see the xylem tissue is really all that green tissue with the big vessels, and then the phloem and bark around the outside. And what we see is when you look at these little markings via laser ablation, you can actually see them, and they're actually visualized. And there's these little marks going through the phloem. They never seem to enter the xylem. And what we're not sure about is if these are potentially the deposited salivary sheets of the lanternflies where they're feeding. Because when they feed, they have a little stylet that they inject into the tissue, like a mosquito, but then they also build this salivary sheath of saliva proteins and carbohydrates that then they can use to explore the tissues. And it looks like potentially after they're done feeding, if that's what we're seeing here, those are being deposited in the stems permanently and potentially damaging the phloem. But we can't really say any of this for certain. We need to further explore it via other microscopy methods. But still exciting to see. It was fun to do this part, and was just kind of cool to see these markings visualized. So then to summarize the overall project, really the main takeaways that we got from these multiple years of study was that lanternflies are successful in altering source sink dynamics and resource allocations with really substantial effects on the roots. Even though they're above ground feeding, they're really limiting root resource allocation. But this was especially in the case of large population sizes. So 100 to 200 lanternflies per vine really drove these effects. And I should mention that our vines were only about 4 to 5 years old. So these results may be different in vines that are 20 years old and much more mature and with bigger root systems and things like that. But again, in vines exposed to small populations, really those extreme carbon limitation scenarios may be avoided, and this could potentially be referred to growers to help influence how they spray insecticides and control spotted lanternfly. And then it looks like the phloem feeding does alter xylem and phloem dynamics, but this is an active area of further investigation. We hope to report more concrete results for that in the near future. So thanks to everyone who's helped me out on this project. It's been a very large team undertaking, and then thanks to all who attended this. So if anyone has any questions, I'll take them now. We have about, I think, maybe like 5 to 8 minutes. So yeah. Sure. Yes. Yeah. Okay. So a question from the audience about whether in terms of the xylem and the phloem dynamics and changes that we saw, if the increase or decrease in activities were influencing the whole trunk size and whether the tissues were, I guess, maybe compensating for each other or influencing the trunk in some way. Right, right. Yeah, so at the whole trunk level, we did see overall trunk shrinkage. And even though the xylem activity was slowing and there was less xylem reduction in size, the phloem was still being reduced too. So there was an overall trunk shrinkage driven by the phloem mainly shrinking, if that makes sense. And I mean, there's some of this data suggesting that the grapevines may be able to osmotically adjust between the phloem and the xylem pretty quickly and actively. And because the lanternflies are feeding on the phloem and removing a lot of those solutes and creating an osmotic kind of differential, there's potential that can also influence things too. But it looks like the trunk diameter is shrinking. So which relative to other literature suggests that it's a stress on growth at least. Yeah. Does anyone on Zoom have any questions? You can either unmute yourself or just submit it in the chat, but other way. Cool. Well, yeah, good stuff. Well, in that case, I guess we'll wrap up a couple minutes early. So thanks to everyone who watched and who's here in person. And yeah, happy Friday to everyone.